Hello and welcome everyone. How are you? I welcome you to the class of biology. So dear students, please get settled in. Please do that fast. And regarding the revision that we had done in the last class, so please, centers, please report your attendance. Please do that quickly. Yeah. Okay. So Shillong, Tamenglong. Hello, good afternoon, Tamenglong students. How are you? Itanagar. Please report your attendance, and we are going to start right away. Okay. All right. So I'm going to give you that PowerPoint presentation once again. So please take a look at what we have done up till now and what we have to do thereafter. So when we talk about cell, okay, so cell is a small living unit. Every organism is made of cell. Okay, so there are two cells shown here. Okay, one is a microscopic paramecium and the other is cork cells, right? So paramecium has a lot of cilia. It's clearly visible in this diagram here. Okay, so Robert Hooke discovered these cells. Okay, and these are basically dead cells. So he called them empty boxes. So he discovered that it is a compartment, so we call it a cell simply. Then I explained to you that as a cell grows larger in size, it has the need to divide itself. It will undergo cell division. We will discover this in the next chapter also, cell division. So you can see a smaller cell has better efficiency because it has a large surface area volume ratio. But if it is a large cell, then it has a large surface area, but compared to the volume, its surface area is quite less. So you can see a large cell has a ratio of 3:1 and a small cell has a ratio of 6:1. Okay? So a small cell is more efficient compared to a large cell. So what is the characteristic of all cells? A cell has a surrounding membrane and it has protoplasm, a thick fluid-like structure, and organelles, and it has a control center in the form of nucleus with its own DNA. There are two types of cells, prokaryotic and eukaryotic cells. Okay, so we are going to discuss this now. Prokaryotic cells are the first cell types to exist on Earth according to the theory of evolution, and the cell types are bacteria and archaebacteria. So in this figure, I have shown you Bacillus bacterium. In this figure, there is a Bacillus bacterium, okay, a small rod-shaped bacterium. It also has a flagellum. So you can see flagellum is coming out from a structure called basal body, flagellum, then DNA is inside, naked DNA, no membrane covered DNA structure. Ribosomes are of 70s type. There is a capsule, there is a cell wall, there is a plasma membrane. Okay, so this is 47,000 times magnified diagram. Okay, it's a scanning electron microscope diagram, 47,000x magnified 47,000 times. Then you get this diagram, right? So prokaryotic cells, no membrane bound nucleus, nucleoid is region of DNA concentration, and organelles are never bound by any membranes, right? Then comes eukaryotic cells. So prokaryotic cell I have already done. I also explained to you that they have phosphate granules, cyanophycin granules, gas vacuoles, and nucleoid, mesosome. All these things I have already discussed, right? So eukaryotic cells, they are bound by membrane. They include fungi, protista, plantae, animalia. They possess many organelles. So eukaryotic cells start from protista. An example of protista is a protozoan. The name is paramecium. So you can see paramecium or the slipper animalcule in this diagram. A representative animal cell. Okay, so animal cell has a lot of organelles that are shown in this diagram. Moving on, a representative plant cell. So I asked you to write the differences between plant cell and an animal cell. I hope you have done it. Okay, so plant cell and animal cell, basically the difference is cell wall, and there are other differences as well. So we will discuss that later. But I expect that you can do this by yourself. Write some differences between plant cell and animal cells. We will come up to this also. No problem. Organelles. What are organelles? Cell organelles are the things which control the cellular machinery. So there are two types of organelles, derived from membrane, single membrane, and derived from bacteria. So we call them bacteria-like organelles. Examples are chloroplast, mitochondria, derived from bacteria. So they are called to be endosymbionts. derived from symbiotic bacteria, ancient association, endosymbiotic theory. So they have 70s ribosomes. They have their own genetic material, just like bacteria have their own genetic material. They divide by simple fission, just like bacteria divide by binary fission, and they are independent. Their division is independent of nucleus. They can divide whenever they want to. Plasma membrane. So regarding plasma membrane, I told you that it contains the cell contents. It has a double layer of phospholipids and proteins. And what is a phospholipid? It is a molecule containing phosphoric acid bound to glycerol, and glycerol is attached to two fatty acid molecules. So it has two tails and one hydrophilic head, right? And it has a lot of proteins. The proteins serve as ion channels. They can also take part in active transport. They can also take part in passive transport. Both things can happen. Okay. So phospholipids, I have already told you, right? So students, I will be, volutin granules are also a type of granules. They are found in some bacteria. Okay. So whenever a bacteria has to store some mineral, Shillong students, they are storing these granules. Okay. So yeah, okay. Yeah, centers, please also report attendance. I believe everyone is online. Nobody has sent their attendance. Please send your attendance fast. Please send your attendance. Centers, please report your attendance. Shillong, Tamenglong and Itanagar, please send your attendance. Okay, 9 students, alright. Okay. Tamenglong, okay. And Itanagar? Alright. Okay, so you are reporting. Yeah, okay, so Shillong students, you are asking me what is volutin granules. So look, volutin granules are basically a type of, it's basically a type of phosphate granule only. Okay, there is nothing different. I have already told you that phosphate granules are basically reserve phosphate sources. The cell stores it because it needs to have a reserve of phosphate in case it decides to undergo cell division. So cell division requires a lot of ATP, a lot of DNA has to be synthesized. So phosphate reserves called volutin granules serve as the storage phosphate material, right? So it also helps in identifying some bacteria. For example, the bacteria causing diphtheria, they are identified by their spatial volutin granules. Okay, so nothing special. They are just phosphate granules. Okay. Okay, carrying on. The protein molecules help in movement across the plasma membrane. So I told you that water, carbon dioxide, ammonia, oxygen, gases and liquids or solvents can move freely, but solute particles cannot move freely. They require the help of proteins. So the proteins are embedded in the lipid bilayer, and small uncharged molecules can move freely. Lipid soluble substances can move freely. Water soluble substances require active or passive transport. Okay, depends upon their concentration. So membrane proteins are of types called channels, transporter, receptors. So we will discuss this later when we in detail when we study transport in plants, transport and movement in plants, right? So there are 4 types. We can discuss this later. Cell walls. Cell walls are found in plants, fungi and many protists. They surround the plasma membrane. Cell wall in plants composed of cellulose. In fungi they are composed of chitin. Cytoplasm, viscous fluid containing granules. Okay, so it contains a lot of storage substances. Alright. So this is visible in this diagram here. Okay. Now one more thing that is visible in this diagram, please look carefully. There are some purple color, blue colored rods in this diagram. They are called microtubules and microfilaments and intermediate filament. Okay. Can somebody explain to me what these structures are for? There are 3 things in this. There are some rod like structures in this diagram. They surround the nucleus, they surround endoplasmic reticulum, and they are present from the plasma membrane to the nucleus. So can you guess what these are for? Can you guess what these are for? Nobody? Well, okay, the thing is, okay, you not answering, okay. Look, have you ever thought about this? Like, let me compare this to your body. Whenever you start walking or you're sleeping on one side, okay, what is the reason that your organs, your heart, your liver, your intestines don't go to the left side or right side? Or when you're running, why does your heart not jump out of your body? In the cartoons, in Tom and Jerry like cartoons, it's very clearly visible that in some conditions Tom's heart comes out. Okay, so I'm not talking about those cartoon like things, but tell me what the reason why our organs, like for example in our body, they're quite heavy. Liver is quite heavy, intestine is quite heavy. So why don't they go on in one direction when you are sleeping on one side and you change your sleeping position to the other side? Okay. No, Shillong students, it is not for transferring energy. First try to answer my question regarding your body. Then I'll ask you about cell. You will be able to relate it better. It's a related question. So can you guess why your organs don't move from their position? They should have moved from the position, right? So for example, your heart could have bounced when you are running or when you are jumping at least, even if your muscles are holding them in place, if suppose, because some students think that muscles hold the organs in place. So if muscles are holding them in place, at some point in time, muscles will get fatigued. At some point in time, only cardiac muscles don't get fatigued ever, because if they get tired, then a person is going to die. But other than that, your smooth muscles are going to get fatigued, your striated muscles are going to get fatigued. Nobody is answering. Okay, try to give it a guess, right? Nobody? Shillong students, would you like to take a guess? Why don't your organs move from their place? Well, like when you are sleeping on one side, they should. Why should not your organs go all to one side? Okay, no idea, I think. Look, every organ is attached to some specific structures called mesenteries. These mesenteries are attached to your body cavity. Inside your body cavity, there are strings, there are fibers that hold each and every organ in their place. If this was not done, then while running or while doing some other activity, your organs would have fallen out. Okay, that would have been the case, but it's not so, because inside their your body cavity, what is happening? The organs are attached with specific string-like structures called mesentery. All right. Now come to this diagram. In this diagram, there are some rod-like structures. These are not for transferring energy or not for transferring anything. These are for providing shape, structure and support to the cell. If these fibers were not there, cell would not have a definite shape. Every organ would have gone at the bottom due to gravity. Look, nucleus is quite heavy, right? Nucleus is quite heavy. So why does the nucleus appear in the center? Why does it not fall down while observing under microscope? What is the thing that is holding the nucleus, mitochondria, every organelle in their place? It is these filament-like structures. It is called cytoskeleton. I will cover this. Okay. So I'm just trying to tell you this. There are three types of fibers, filaments, and these filaments are part of cytoskeleton. It is called intermediate filament, microfilament, and microtubules. These hold all the organelles in their proper position. All right. So this is cytoskeleton, microfilament, microtubule, intermediate filament. It provides mechanical support, anchors organelles, and helps move the substances. Okay. So this is a diagram showing the microtubules and the other structures present in the cell everywhere. I will also give you another diagram of when the fibers are seeing sparkling against the light. So you can see that the network of fibers is holding everything in place. Okay, cilia and flagella. Cilia and flagella we have to do today. Okay, so cilia short, used to move substances outside human cells. So inside your bronchial tract, okay, respiratory passage. Do you wonder why we sneeze, why we cough? It is all because of the action of cilia. If the cilia are unable to remove any foreign particle, you get a sneezing reflex or a coughing reflex. Cilia remove the unwanted bacteria away from your respiratory passage. Nothing should get inside. So it is covered with mucus, and this mucus is given a definite direction to flow upwards. And this waste material comes out from your nasal passage from time to time. Everybody knows what it looks like, and there are some people among our friends also, like everybody knows some people who will do this act in the front of everybody. They let them know that they are removing some waste material from their nasal passage, and they will want everyone to know. Okay, so I am not taking any names here, so you know better who has this habit. So I had some friends who had this habit of doing it in front of everybody and not getting ashamed of anything, not doing it in private. Anyway, so cilia has this function, right? So cilia is present in a lot of structures. Whenever something is needed to be moved inside the human body, a fluid is decided to be moved from one place to other, cilia will help in it. Okay. So inside our bronchial passage, there are ciliated cells which help in this act. Flagella are whip-like extensions, and they are found on sperm cells in human body, and their flagella are normally found in lower organisms also. Okay, they are always arise from basal body like centrioles. Then cilia flagella ultrastructure. It contains a 9+2 arrangement. Okay, so this we will discuss later. Centrioles, I will discuss this also. It helps the cell in dividing, and it forms a spindle apparatus. Okay, membrane bound organelles. Nucleus is a membrane bound organelle, and it is a double membrane bound organelle. Control center of cell. Okay, nuclear envelope. DNA is to be discussed in detail in biomolecules, and nucleolus. Okay, so this all part, the remaining part we will do today. Endoplasmic reticulum we have done yesterday. So it's a transport network consisting of the network of interconnected tubules. It is of two types, rough and smooth. Rough has ribosomes. Smooth does not have ribosomes. It helps in lipid synthesis. Rough helps in protein synthesis. Golgi apparatus involved in packaging of the substances manufactured in the cell, normally by the endoplasmic reticulum. So it's a processing and packaging center. Okay, so Golgi apparatus helps in forming vesicles. Vesicles are fused with the Golgi membrane, and the molecules may be modified by Golgi apparatus. So this is the GERD apparatus, Golgi body endoplasmic reticulum lysosome system I told you about. Okay, so this is the pathway in which the things happen. The green pathway represents target molecule synthesized from rough endoplasmic reticulum, transported to Golgi body, forming secretory vesicles, and secretory vesicles thrown out of the cell. Okay, so there will be secretion from the outside to the outside. Then there are lysosomes. Okay, lysosomes are containing digestive enzymes. I have discussed lysosomes yesterday. It is having a very thick membrane. So they help in cell renewal, break down the old parts, digest the invading organisms. For example, in the case of white blood cell, it's having the capability of digesting the bacteria. So it is due to lysosomes. Vacuoles are very large structures. They occupy about 90% part of the cell. Okay, so it's a very large structure filled with cell sap and some excretory material also. Bacteria-like organelles are two now: mitochondria and chloroplast. So this we will have to do today. So let's start from where we left yesterday. Okay. So lysosomes was the part where we had done till yesterday. So we had completed girdle apparatus or endomembrane system. So today we will be dealing with other cell organelles. Okay. So I believe you are ready. I believe you are ready, right? Okay. So let's begin. So all the organelles that we have covered up till now, they were single membranous organelles. Now we will cover some other organelles like the power producing, the energy producing organelles called mitochondria. It's a very important organelle. So let's cover this first. Let's cover this. So what is a mitochondrion? What is a mitochondria? A mitochondria is a double membrane bound cell organelle, and it's the powerhouse of the cell. Without it, the cell cannot have any power. One more interesting thing that I want you people to know is that mitochondria has its own genetic material. So it is called semi-independent. It does not depend upon the nucleus for it repairing its proteins, repairing the membrane, or making enzymes also. So everything is manufactured inside. So why does not the bacteria like mitochondria become free of the cell and start doing everything by itself? Why does it not happen? Because it has its own genetic material. So you can suppose that because it has its own genetic material, it is like equivalent to a small bacteria. So why does the mitochondria hesitate in doing so? What is the reason? Why can the mitochondria not become independent and become a separate living entity like a cell? Can you guess? Why should it not become totally independent? Outer membrane. Inner membrane. Inner membrane is having folds, so it is folded. (70S) ribosomes. Okay, and then we have, this is the genetic material. Okay, it is double stranded circular covalently closed DNA. It's DNA, right? Now there is one more interesting thing here. On the inner membrane, there are some small lollipop like particles and these are called oxysomes. They are present all over the inner membrane. I am drawing only some. Okay. So this is an oxysome. Basically, wait a second. Let me draw it. There is no need of drawing it with separate color. Yeah. So this is the FOF1 particle or the oxysome. Okay, FOF1 particle or the oxysome. Okay, and these are present all over the inner membrane. So please draw it quickly, and then we will have to write some things about this. Meanwhile, I hope you have found the answer to my question. Okay, so please finish this quickly. Alright, so we have, okay, done. Alright, so let's write a few things about this mitochondria now. Mitochondria are double membrane bound cell organelles with inner membrane having multiple folds, and bearing FOF1 particles. Okay, they look like a tennis racket. FOF1 particles called oxysomes. And oxysomes, what are they important for? They are having ATPase activity. ATPase is anything which is able to synthesize ATP. So ATP synthesized with the help of these particles. ATPase activity. Okay. So oxysomes have ATPase activity inside them. They also have their own genetic material and this genetic material is double stranded circular DNA. Double stranded circular DNA. Double-stranded circular DNA. Okay. So we call it DSCCC, double-stranded circular covalently closed DNA. Okay. So we call it double-stranded circular covalently closed DNA. I believe I have already told you this many times, so that is why I am not repeating it. Double stranded circular covalently closed DNA. Okay. All right. So one more thing is mitochondria number. It is highly variable. Mitochondria number depends upon how many organelles does this cell require. If this cell is going to produce very much large amount of energy, then we need to produce a very large amount of mitochondria. For example, pigeon in the breast muscles or the pectoral muscles, where Krebs cycle was first discovered, in those cells, in the pigeon's muscle cells of chest region, the number of mitochondria is very, very high. Okay, compared to muscle cells of our body, they will be very high. But our muscle cells will have more mitochondria compared to some other cells such as pancreas cell or intestine cell. Okay, So some cells need to produce more energy. So when they need to produce more energy, the number of mitochondria will also be more. So please write, number of mitochondria is dependent upon number of mitochondria is dependent upon the metabolic activity of the cell. The metabolic or physiological activity of the cell. Metabolic or physiological activity of the cell. Okay. All right. One more thing now. Look, there is a term that is going to be repeatedly used from now on. It is called matrix. Matrix refers to the liquid substance that is flowing inside the internal side of the mitochondria. Okay. So please write, the inner compartment of mitochondria, the inner compartment of mitochondria is filled with a homogeneous substance called the matrix. Homogeneous substance called the matrix. Called the matrix. Okay. All right. Now one more thing, the last point. Mitochondria are semi-autonomous or semi-independent organelles. They are semi-autonomous, or semi-independent cell organelles. Semi-autonomous or semi-independent cell organelles. Why? Because they have their own genetic material. They have their own genetic material. Okay. Their own genetic material, it is called nucleoid or genophore, but it's not necessary to write it. It's double-stranded circular covalently closed DNA, own genetic material, and 70s ribosomes. This means they have the capability to synthesize proteins by itself, and it happens also. Mitochondria synthesizes its own proteins. So they are called semi-autonomous, semi-independent organelles. They are not dependent upon the nucleus. Normally for other cell organelles, when the cell is going to divide, the organelle is also going to divide. But in this case, because these are semi-independent, they divide on their own by fission. They do not need the nucleus to help them divide. They divide on their own by fission method. On their own by fission method. mitochondria, they can basically symmetrically divide and they can form new cells, form new mitochondria. So my question to you is, why does the mitochondria not become independent by itself? What is the reason that they don't become independent by itself? Any idea? Okay. The reason is because mitochondria requires a lot of energy rich substrate. It requires glucose, it requires other materials, and mitochondria cannot absorb these things by itself. So it is dependent on other cell organelles also, but for its own requirements, it can be self-sufficient. For its own requirements, provided it is given with certain substances, but it is not fully independent, so it is somewhere in between. And that is why we call it semi-independent. It divides the fission method or binary fission method. So this is also the similarity with the bacteria. This is also the similarity with the bacteria. If the mitochondria and the nucleus are both able to produce 70s ribosomes, won't there be an axis of it? No, there is not going to be an axis of it, because in the class 12th you have a chapter called molecular basis of inheritance. In that unit there is a last topic called the gene regulation, Lac-Operon model, and also that. Thing is, we can make proteins, but we can make proteins only up to a limit. Okay, and this limit is decided by the environmental factors or other factors, or the enzyme availability. So we are going to decide, the organism is going to decide how much to make, and it cannot waste resources also. It will not waste the things also. So unless and until enzyme is required, it will not be made. If it is made, it will be in a proper proportion. So there won't be an axis of it. If the cell finds that there are axes of proteins accumulating, it has everything in axis, it will simply undergo cell division. It will make a new cell. The other cells will also start making proteins. So a lot of bacteria will be produced in a lot of time, but in normal cells this does not happen. Why? Because mitochondria are not bacteria. Mitochondria are not bacteria. They are not independent. The nucleus will decide how much food materials I am going to take. Okay, so mitochondria are not independent. All right. So mitochondria, you know, in the chapter called respiration, we will deal with mitochondria. Mitochondria play an important role in respiration. Mitochondria play an important role in aerobic respiration. Play an important role in aerobic respiration. Role in aerobic respiration. Aerobic respiration. Shillong students, the one at the front, I believe you have to start taking notes. I know what you are doing. Please start taking notes, okay? I can see it. I don't want to speak out, okay? So please start taking notes. I can see what you are doing, okay? So your phone is getting charged and so and so. So I believe talking is important, but there you will get a lot of time for it, okay? All right. So let me continue. So everybody is, I understand everybody, you are teenagers, so it's important to talk, it's important to do what you want, but please also take care of your studies, okay? I'm fine with it, but please take care of studies. All right, so what are plastids? Plastids are found in all plant cells, found in all plant cells and euglenoid organisms, and euglenoids and euglenoids. Okay, so these are very large structures. Actually, a chloroplast is somewhat larger than mitochondria, so they are very easy to observe. Okay. They are large and can be easily observed. They are large structures and can be easily observed under microscope, and can be easily observed under microscope. Okay. So, everybody has seen chloroplasts, I believe, in some form or the other. You must have seen guard cells, stomata. Okay. So, you must have seen all these structures. Okay. So, plastids are very easily observable, right. Yeah. So, if you have a microscope in your school, you can see green dot like structures. You might not be able to see the very extremely magnified structure, but you can see green dots and they are very easily identifiable. Okay. So, you can see these green dot like structures, and if you have a microscope which is having about 500x or more magnification, about 1000x, then you might be able to see some details in the chloroplast. Okay. So, now plastids are of three types. Plastids are of three types depending upon their pigment composition. Plastids are of three types based upon types of pigments present. Types of pigments that are present. So, what are they? Number one is chloroplasts. I will write chloroplast with a green color so that it is clearly understood. Chloroplasts. Second one I will write with a different color. Let us take red color because yellow is not clearly visible. Chromoplasts, color containing plastids. Third is leucoplasts. So, leucoplasts are basically storage granules. So, they can store fats, proteins, so many things. Leucoplasts. Okay. So, what do chloroplasts contain? Contain chlorophyll and carotenoid pigments. Carotenoid pigments. Because carotenoids are yellow, orange, they are of carotenes, xanthophylls, so many things. Okay, carotenoid pigments. Okay. Now, what are chromoplasts? Chromoplasts do not contain chlorophyll. Otherwise, other than that, they will contain the full spectrum of carotenoid pigments. They contain carotenoid pigments. Carotenoid pigments are contained in there. Okay. So, chloroplast separate thing, chromoplast separate thing, because one has chlorophyll and the other does not have chlorophyll. Next, come on to the leucoplast. Leucoplasts are not having any kind of pigments. Okay. Pigments are absent. Pigments absent. So, they are colorless plastids. They are colorless plastids. colorless plastids. So they are of three types. They store nutrients and are of three types. They store nutrients and are of three types. First, it is A type, amyloplasts. Amyloplasts. Okay. They store carbohydrates or starch. Carbohydrates or starch. Most starch is the most common carbohydrate. That is why I am writing it. Second, B. Example you can write potato, rice, etc. Second is elaioplasts. Elaioplasts. They store oils and fats. They store oils and fats. Okay. Can you give me an example where oils and fats are stored? Mustard seed, sesame seeds, groundnut seeds. Alright. So all these conditions where oils are stored, they will be under elaioplasts. Number 3 or C, three types, right? Then the number 3 is aleuroplasts. Aleuroplasts. Can you give me an example of aleuroplasts? It is derived from aleuron layer in monocots. So aleuron layer will have aleuroplasts. It is going to store proteins. Wherever proteins are stored, they are stored in aleuroplasts. They have protein granules. Okay. All right. So these are the three types of plastids. Now let us draw the structure of chloroplast. Chloroplast. Chloroplast is a double membrane bound structure also, but here the inner membrane is not having any folds. The inner membrane has already formed some membranous bag like structures, and these are called thylakoids. Okay. Thylakoids. Thylakoids like this. Okay. Thylakoids like this. Like this. Like this. Now, what is a thylakoid? Thylakoid is this structure. This stack of coin like structure, it is thylakoid. And one coin out of it, it is a granum. this coin like structure, this is a granum. This structure is a thylakoid. Okay. This part it is called stroma. These are 70S ribosomes. Genetic material, which is DNA. This is the outer membrane. This is the inner membrane. Inner membrane is quite smooth, right? Okay. So this is the structure of chloroplast. Structure of chloroplast. So it is also a semi-autonomous organelle. You can write the same things about it, just as you write about mitochondria. I can see some people are writing very, very less. Yeah, I can see everything, including who is typing on their phones and what not. Okay, so please don't do it every time at least, okay? Please don't do it every time. Maybe you have somebody's birthday today or somebody else, something else. So you're not writing. If you're not writing, I can see. Okay, that's okay. I'm cool with it, but please don't miss my notes. And when I ask something, please try to answer. All right. So please look here. This is a double membranous structure. You have seen the structure of chloroplast. So you know what it is like. It's having a huge stack of these coin-like structures and one below the other. And this structure here, it is connecting to stacks of coins together. It is called stroma lamellae. Stroma lamellae. Okay. It is actually a sheet-like structure. And what is this granum? Granum is a single coin-like structure. Imagine having one on top many coins, and this becomes a thylakoid. So there are two stacks of thylakoids that are connected together by stroma lamellae. A very important fact: where is chlorophyll found in chloroplast? Where is chlorophyll found in chloroplast? Can you guess? Where do you find the chlorophyll? I have not shown you here where you can find the chlorophyll. So where can you find the chlorophyll? Mesophyll. Mesophyll is a type of cell. Inside mesophyll is chloroplast. I am asking you where inside the chloroplast you will find it. It's a structure inside mesophyll cell. So now you have to tell me where inside the chloroplast can you find the chlorophyll. Mesophyll answer, your answer is good. You can find chlorophyll in mesophyll cells. But where? In the chloroplast. Where in the chloroplast? Yeah. Thylakoids contain chlorophyll. Stroma does not have any chlorophyll. Only these bag-like structures, coin-like structures. These coin-like structures are nothing but bags made of membrane. So please write now about chloroplast. Chloroplast has two membranes surrounding it. Both inner and outer membrane are smooth. They are smooth membranes. The matrix of the chloroplast is called stroma. The matrix of the chloroplast is called stroma. Is called stroma, and inside the stroma are membranous bag-like structures called thylakoids. In the stroma, are present membranous bag-like structures called thylakoids. Each thylakoid consists of multiple grana. Each thylakoid contains coin-like structures, membranous bags, each bag is called granum. Okay. The chlorophyll pigment is stored inside the thylakoids. The chlorophyll pigment is found only inside thylakoids. Only inside thylakoids. Okay. So this is about chloroplast. They have 70s ribosomes. And independent nuclear or genetic material. Nucleoid and genetic material. Independent nucleoid and genetic material. So basically this is one and the same thing. You can just write independent genetic material. So they are also considered semi-autonomous organelles. Semi-autonomous organelles. Okay. So do you need a break now? 5-minute break, I believe. Some people, yeah, raise your hands if you want a break. Okay, no, alright, you want a break? Okay, take a break of 5 minutes. Okay, yeah, please take a break of 5 minutes, and then we'll start with ribosome and nucleus, etc. Okay, so come back after the break. Okay. Okay, so so we are back now. So we had covered that chloroplast is a semi-autonomous or semi-independent cell organelle, and it contains thylakoids, and these thylakoids contain chlorophyll. It contains 70s ribosomes, it contains genetic material, and it has two membranes. Okay, so photosynthesis takes place inside chloroplast. Please write. Okay, please write. Photosynthesis takes place inside chloroplast, right? So this is all about chloroplast. Now please put the heading ribosomes. Ribosomes. Okay. So regarding ribosomes, what do ribosomes serve the function of? Ribosomes are the protein factory of the cell. Ribosomes are the protein factory of the cell. Protein factory of the cell. So they synthesize proteins, but my question to you is, what is ribosome made of? What is ribosome made up of? Nobody? Okay. Ribosome is made up of proteins plus ribosomal RNA. Okay. So the thing is, you can imagine that ribosomal RNA, it is attached to some protein like structures. Okay. It is attached to some protein like structures, and these are the structures which make up the ribosome. So what happens is, there is a large coiled network of ribosomal RNA, and it is coupled to proteins, and this together makes the ribosome. But the important structure behind this is, it is having a network of magnesium ions. It is having a network of magnesium ions, and it is having a structure which is partly credited to the magnesium ions, which helps in holding these things together. So please write. Ribosome is made of, ribosome consists of two subunits, small and large subunit, held together by magnesium ions. So if magnesium is present in the correct concentration, it is going to hold these two units together. If magnesium ion concentration is removed, it is lessened, in that condition both the subunits will become separate from each other. Okay, so now you can draw it. There are two subunits, small subunit and the large subunit. Small subunit and the large subunit. Okay, so the small subunit, in the case of 70s ribosome, it is made up of 30s particle, and in the case of large ribosome, the eukaryotic ribosome, it is made up of 40s subunit. So you can write now. In eukaryotes, the ribosome found inside the cell, ribosomes found inside the cell are of 80S type, and it is made up of 60S subunit plus 40S subunit. 60S is larger, 40S is smaller. Okay. All right. And in the case of prokaryotes, the ribosomes are of 70S type. The ribosomes are of 70S type. Are of 70S type. So when we talk about 70S type ribosomes, it has two subunits. These are 50S plus 30S. Now there is one more important thing you should note here. There is no mathematical relationship between these two subunits. For example, if you add 60 plus 40, you get 100, not 80. If you add 50 plus 30, you get 80 and not 70. So what is the reason? Do you know the reason? Okay, the reason is there is this S value, the number S denotes sedimentation coefficient or Swedberg unit. It does not have a direct mathematical relationship with the size. Okay, it gives an estimate that something is bigger, but it does not have a direct mathematical relationship with the size. So you can write now, S denotes Swedberg coefficient, Swedberg unit, or sedimentation coefficient. Sedimentation coefficient. So do you understand why Swedberg unit, or what is the meaning of it and how it is calculated? Okay, I believe you don't know. Okay, what happens is, there is a large, actually I think I should draw the diagram for you. What is S? So regarding S, I have to explain this to you, then only you will be able to understand it. So I am going slightly off topic here. S, sedimentation coefficient, how is it calculated? So you need to understand something about sedimentation coefficient. Okay. So it is calculated using centrifuge. It is calculated using centrifuge apparatus. Centrifuge apparatus. Now what is the centrifuge like? So let me tell you this. Centrifuge is like a machine with having, these are two test tube holders, they are attached to a single rod and it can rotate in clockwise or anti-clockwise direction at very high speed. These are test tube holders. Test tube holders. Inside the test tube holder you can put two test tubes, and both these test tube holders are attached to a long rod, and this long rod can be made to rotate at very high speeds. One more thing, these test tube holders, they can also become horizontal. Right now they are vertical, but they can also become horizontal. So when they become horizontal, it will be like this. So they have a hinge, they can rotate about that hinge, so they can move from this position to that position. Alright. So what happens is, inside these test tube holders we are going to put two test tubes. We are going to put two test tubes, and these two test tubes are connected, okay, by a single system, by a single rod, and I am rotating this at a very high speed. So when I am going to increase the speed, these test tubes will become horizontal. It will be dependent upon the function, the speed. So these test tubes, they contain some material. So what I am doing is, I am preparing some chutney. I am preparing some plant chutney, okay, and I am taking this chutney and I am putting in these jars, okay. So what is this chutney or the plant extract contain? The plant extract contains the remains of the cell. I am taking the cellular components, I am beating them together, and I am separating one cellular component from the other. I am breaking the cells. Okay, when I am making a chutney, I am making a plant extract, I am making basically, I am put them plant material in a mixer, I am basically breaking down the cells. So I am going to get a lot of cellular components in my extract, this green colored structure, okay, green colored liquid. It is going to contain nucleus, ribosomes, endoplasmic reticulum, Golgi body, lots and lots of cell organelles. When I rotate it at a high speed inside the centrifuge, it will experience a force which is many times more than the gravitational force. Many times more than the gravitational force. So on the bottom side here, the test tube, it faces a force called the centrifugal force. So have you experienced a centrifugal force at any point in time? If you have been to a fair, okay, when there are large fair rides, okay, Ferris wheel like structures, and you go up to the wheel. So you experience a certain position in time when it starts rotating, and you sit on the giant Ferris wheel and you rotate some force, right? And you experience some rotational force, okay, and then you also experience as if you are going to fly away from the circle. Everybody remembers it? Okay, that is called centrifugal force. Yeah, exactly. Yeah, on the Ferris wheel you can experience this kind of sensation. Now this is a similar kind of arrangement. It will also experience centrifugal force, and this centrifugal force will be directed away from the center, and because the test tube has become horizontal because of high speed, so the test tube will experience a maximum force on the bottom side. So the components which are most heavier, for example the nucleus, it will settle first of all. The components of the cell which are the heaviest, they settle first. So this is called sedimentation. So what I am going to do here, I am allowing the centrifuge to operate at very high speeds, and at very high speeds and for considerable length of time. First of all, nucleus will separate. After that, endoplasmic reticulum, Golgi body, heavy cell organelles will be separated. At the very last moment, what I am doing is, I am separating the cell organelle and I am taking the filtrate, and then I am putting it again for longer duration of time, then Golgi body will separate, then endoplasmic reticulum will separate. So, heavier components separated first, lighter components will be separated later. This is the principle of centrifuge. And if it is separating earlier, I will give it a high sedimentation coefficient, because it is heavier, it is denser, it is having high density. Nucleus has a sedimentation coefficient value in about 10 to the power 5 or 10 to the power 6. Endoplasmic reticulum and Golgi body and mitochondria has lesser value. At the last, ribosomes will be separated, so it is given the least value. So, which is heavier, 80s or 70s? 80s. 70s is slightly less heavier, it is slightly lighter. And if you change the magnesium ion concentration, ribosome will also separate into different subunits. So, we can separate subunits also, we can separate ribosomes also. Sedimentation coefficient gives an value, gives an idea for how much time we are rotating the centrifuge and at how much speed. Multiply these two together, you get sedimentation coefficient. So, it has no direct relation to the molecular mass, it has a direct relation to the speed of rotation and time of rotation. Molecular mass is not coming here. So, even though 80s ribosome is made of 50s and 30s, if you add them together, you will not get 70s, sorry, 70s or 80s. 50s and 30s gives 80 value, not 70. So, that means there is no direct mathematical relationship. It gives an idea of how much heavier substance is, but there is S has no direct relationship with the molecular mass. So, you can write now sedimentation coefficient gives an idea, sedimentation coefficient depends upon speed of rotation and duration of rotation. Speed of rotation and duration of rotation. Not to the molecular mass. Okay. So, nucleus, for example, has a very high sedimentation coefficient. Nucleus has a very high sedimentation coefficient value. Sedimentation coefficient value, because it separates first, value because it separates first. Separates first. Okay. But other organelles, they have lesser value. Ribosome is separated the very last, so it has a least sedimentation coefficient. Ribosomes are separated at the very last, so they have least S value. Are separated at the very last, so they have least value of sedimentation coefficient. Least value of sedimentation coefficient, right? So, now you get the idea. why and how S is calculated. So, this was just to give you an estimate of the sedimentation coefficient and its importance. Okay. So, I have placed this in a separate box so that it becomes clearly distinct from the other things. All right. Okay, so now you've finished it. All right. So, let me tell you further things. Ribosomes manufacture proteins with the help of with the information from messenger RNA. Yeah, I understand it's time. I am just leaving you. Okay, I understand. So, in class 12 syllabus, when I am going to cover class 12 syllabus, I am going to show you these things. For example, let me show you one. Just a second. Okay. So, yeah. This is a structure of ribosome. Okay. Small subunit is like this, large subunit is like this. Large subunit has two transfer RNA and amino acid binding sites, and they will help in reading the structure of the amino acid that has to be decoded. So, this structure, it will become clear when you study the class 12 syllabus, and in that I will give you the full details. So, you can just get a fair idea looking at this. Okay. So, the messenger RNA is read on the ribosome. Okay. So, transcription, translation, all those processes where the translation process happens. Okay. So, this is the figure how amino acids are interacting and making new peptide bonds. Okay. So, this will come in class 12 syllabus. So, in due time I will take this up. Okay. So, right now you can just get an idea that ribosome makes amino acids attached to each other. It makes proteins basically, and this is done with the help of small subunit, and the large subunit is going to form the protein bonds. Okay. So, coming back, you can draw a small diagram. You can draw a small diagram regarding protein synthesis. Small subunit is going to read the messenger RNA. Messenger RNA is read by the small subunit, and the transfer RNA comes inside these two subunits. Okay, these are two places. This is large subunit. The peptide bond formation takes place inside the large subunit. Messenger RNA enters the small subunit. In protein synthesis, messenger RNA is read by messenger RNA is read by small subunit, while large subunit approaches transfer RNA and forms peptide bond. Okay. So, this is all about ribosome today. A few things, small things remain. Structure of structure of nucleus, heterochromatin, euchromatin, and then comes the structure of chromosomes. So that last part will be dealt in it will take about 1 hour in the next class. So thank you, dear students. I hope this has been informative for you. This full PowerPoint presentation, it is not complete. I'll complete it today, and then I'll send it to you on the group. Okay, so you can take it from there also. And those who do not have the PPT, if you have the WhatsApp, let me know. I'll send you these notes as well. Okay, these notes I will send you these as well. Okay, thank you for your time, and I'll see you in the next class next week. Thank you.